So in lab 4, you are going to be testing for the 4 different classes of our biological molecules. So in order to do that, we need to use what are called control groups. So this is really your first lab this semester where we actually have to use control groups. So why do we need controls, right? Why is this important? So if you look back at lab number 1, where we talked about the scientific method. Remember, we talked about experimental groups versus control groups. Basically, why we need control groups. 2 big reasons. 1, we need a comparison. We need to know what we're looking for and what that's going to look like. We need to be able to know that the thing we're actually testing for is being tested for, and so we can make both qualitative and potentially quantitative comparisons. The other thing we need to do is verify that everything is working correctly, right? Did the experiment actually work, right? Was there a difference between the control groups and the non-control groups? And the other major thing we use it for in this lab is we're going to use 2 different types of controls. We're going to use what's called a positive control and a negative control. And if the 2 control groups come out looking exactly the same, the experiment was not done correctly, right? It's a major checking point for us. We know that something went wrong, whether we didn't do it correctly in human error, or there's maybe something wrong with the solutions that we're using. Like, there's so many things that could cause an experiment not to work. And this is a good place for us to check ourselves, make sure that we are actually looking at an experiment that something happened in. So if they're the same, something's wrong and we need to fix it, right? So let's look at the difference between a positive control group and a negative control group. So a positive control group is going to have what we are looking for in it. So in this lab, we're going to be testing for biological molecules, right? So a positive control is going to have that molecule in it. So for example, if we're looking for starch, we will have a positive control that is starch. It will literally just say starch. So that we know when we run that test what it should look like, right? So in this case, we're going to be using colors, and we will then know what color the test tube should be turning based upon what our positive control gives us. Then the negative control is going to not have the molecule in it, right? So again, if we're testing for starch, we're going to use something that we know does not have starch in it. Now in this lab, we're going to be using water for every single test, right? So water is a great negative control when you're talking about biological molecules because it does not have any 4 of those biological molecules in it. But water is not always the negative control. So don't get it in your head that water is the negative control for everything. It's not, right? It really depends on what the experiment is. So later in the semester, we'll actually use a control that is alcohol-based and not water-based because of the experiment. So the controls depend on the experiment that we're running. In this case, because we are looking at biological molecules, our positive control will have the biological molecule, our negative control will not. So in this lab, what you are going to be testing is a bunch of things you don't know. These are called unknowns, right? Which should make sense. So we're going to test things to look to see what is inside of them, right? Do they have proteins? Do they have DNA? And we'll be testing. Now, for some of these things, you might be able to make a hypothesis. You might be able to go like, oh, well, we're testing an egg, so I've heard eggs have protein in them. So, I mean, they probably have protein in them, right? That's a hypothesis, right? You're using your knowledge to make a, general guess that, yeah, eggs have protein in them. And then you're going to test it to actually confirm it, right? So even if you can make a hypothesis, that doesn't mean that it's, not an unknown. You still aren't sure. You still have to test it and confirm it. So the unknown solutions, including egg, will be something that you can often form a hypothesis about. Now in this lab, you will also be given a flat out unknown. It will be unknown 1 or unknown A, depends on how they're labeled each semester. So that you will have no idea, right? So you will have to test it for all of the tests that you're doing and actually determine if the unknown has a specific biological molecule in it. And then you can potentially make a guess as to what the unknown actually is, right? And we use foods for this, so you'll have a list of foods that you'll compare it to. So this is also a good place to talk a little bit about quantitative data data versus qualitative data. In this lab, we are going to be using qualitative data. You are going to be looking at colors and descriptions of certain reactions. You are not going to be really measuring anything. So there's no way to actually put a number on it, right? So quantitative data is a number, right? Maybe you're measuring it in millimeters, or my example here in minutes. You are going to take specific measurements that will give us number data that we can analyze. Qualitative data is more about looking at what happened and then applying what we should understand. So with qualitative, it's going to be even more important, in my humble opinion, that we have strong positive and negative controls for comparison so that everyone is making the same conclusions. So in this lab, we are going to be doing 5 different procedures. We are going to start with carbohydrates. So carbohydrates is our first major biological molecule. Remember that it has monomers as well as polymers, right? So carbs are our sugars. So monosaccharides are the monomers. They're simple sugars. So most of you have heard of, glucose, fructose, sucrose is a disaccharide. And deoxyribose is actually the sugar in DNA, is considered a monosaccharide. So these monomers can then get put together to form our polymers. So in a dehydration synthesis reaction that we talked about in the first video, we can take these monomers, put them together, pull water out, and form a polymer. So in this case, in the carbohydrates, we have disaccharides, which have 2 of the monomers that are linked together with covalent bonds. Really? I'm not the anatomy teacher who doesn't know how to type. I'm sorry. That should say bonds, obviously. Not the anatomy teacher. Okay, I'm totally the anatomy teacher. So disaccharides are going to be 2 sugars put together, right? 2 monomers put together. Sucrose is an example. Lactose, which is found in milk products, is a really great example as well. Now, if we put more than 2 of our monomers together, then we will form what's called a polysaccharide. See, I wrote bonds correctly when I typed it on this line. These are also going to be put together using dehydration synthesis in order to form our covalent bonds between the monomers. So these are things like starch, which is actually the experiment you're going to be running in this lab. Glycogen is another great example, which is an animal polysaccharide that's stored in our livers. Cellulose, which is basically dietary fiber used for cell walls in plants. And then chitin, which is a structure found in insects that have major exoskeletons. They're very strong. So in this first procedure, procedure 4.1, you are going to be looking for what are called reducing sugars. These are simple sugars that are losing electrons, and this brings in a full conversation about oxidation and reduction. So as you're moving through lecture, you're going to hear about redox reactions, which are reductions and oxidations, where one thing gains an electron that's given up by another. All right, so when one thing is oxidized, another will be reduced. So you will talk much more about this later on. Our reducing sugars are those that give up the electrons, which is going to make them react really well with the test that we use. So the test that we're going to use for 4.1 is called the Benedict's reagent. So the Benedict's reagent is going to get added to your solutions. So in procedure 4.1, in table 4.1 in your lab book, you are going to see all of the solutions you're going to test. That's your unknown, at least in this sentence right here. So you're going to add 2 mils of it, 2 milliliters of the Benedict's solution, and then you're going to throw it in some hot water. All of these do have to be in hot water for a minute, and what you will see then is that the test tube will have time to react, or should I say the solution will have a chance to react with the Benedict's solution to find the reducing sugars. Then you're going to remove it and you're going to view it. Now remember, hot things are hot, so be really careful there. But what you're really going to do here is compare each one of the test tubes. So in this one, it's 7 test tubes. You're going to compare the 5 unknowns to the controls. So in this situation, your positive control is the one for reducing sugars, which is test tube number 1, and your negative control is going to be water, which is test tube number 2. So you're going to be comparing the test tubes 3 through 7 to test tubes 1 and 2. Does it look more like test tube 1 or more like test tube 2? So to give you an idea of what this will look like, and yours might look different because there might be more or less sugar, but these are actually the two controls. So this first one is an orangish, almost sometimes turns like a rusty orangey color. This is a positive reaction for the Benedict solution. The blue is a negative because the Benedict solution is actually blue, right? So this means there's no sugar in the solution. This would mean there is sugar. Now again, remember that there's a range, right? There's a range of how this color is. For example, even if it's a greenish color, right, it has moved from blue to a little bit of sugar, right, and that would mean it's slightly green color. So again, there's a range. This is why it is considered qualitative data, right? It's based upon our judgment of color. So now let's look at starches. So starch is a polymer of glucose, again, formed from a dehydration reaction, right? That is put together to form this strong polysaccharide. So in order to test for polysaccharides, we use iodine. All right, now when iodine finds starch, it turns purple, which you actually also should have seen in your cell lab when we were looking at potatoes, right? We looked at amnioplasts, which contain starch. We used iodine as our stain so that you could find the starch. Same will happen here. You're going to take your solution, you're going to add 0.5 milliliter of iodine into each one of the test tubes, and you're just going to view the color. You don't have to boil this, you don't have to do anything, you're just going to swirl it and voila, you will have a reaction. And again, you're going to compare to your controls, comparing to your starch control and comparing to your water control to figure out is starch present or not. So when we look at these test tubes, you will see that this is basically black. Depending on how much starch is here, it will be purple. It just has to have enough light to go through it to be purple. This is a purplish black color. So the lighter the purple, the less starch that's present. The darker the purple, more starch is present. And then you will have the negative control, which basically just looks like iodine in water, right? It's a ambery type color. That would be a negative reaction. And then in your table, you will put your observed color and then was it positive for starch or negative for starch.